Good morning, everyone. Today we will have our natural product session, and I first of all would like to welcome all of you, the audience, but also the speakers. And this natural product session was organized by the IUFA Natural Product Committee, and we are very happy to have this session. It will give you an overview from basic targets of natural products to preclinical up to clinical studies, and you will see that natural products is a fast-moving field. And we will have, I will give my hand to Professor Spedding, who will introduce the first speaker.
Okay, thank you. Well, actually, it's a considerable delight for me to be able to introduce Professor Jean-Luc Wolfender, who has been one of my guiding lights since I was Secretary General of International Union of Pharmacology, because I believe that metabolomics is probably the best omics at present for our major issues. And Professor Jean-Luc Wolfender is very famous in the field of natural products. He's a chemist. He's also Dean of the Faculty of Sciences at the University of Geneva. His work is, for me, actually one of the key areas forward for natural products. And he's going to talk today about the combined target isolation and metabolomic approaches for the efficient discovery of potential therapeutic agents from natural sources. And this gives us a possibility of being able to deconvolute natural products and also to be able to effectively work out what's potentially active and what's potentially toxic. So, Jean-Luc.
Thank you very much, Michael, for the very kind introduction. So it's a pleasure to be here at IUFAR. And indeed, I will try this morning to show you a little bit what we do. Mainly, I'm a chemist, so I will show you mainly what we do on the technical aspects and on the way to apply metabolomics to try either to annotate compounds in complex mixture, herbal extracts, for example, but also use this technique in order to target the isolation of bioactive compounds for drug discovery. So, just a brief introduction on natural products. Natural compounds are looked at privileged structure. They have been shaped through evolution, mainly as defense compounds, but they are heavily used in traditional medicine, in herbals, for example, and this has proven their activity against many targets. And this is mainly related to their, chemical complexity, which allows very selective binding. And if you look at these natural products, you can summarize the diversity of this compound in a kind of chemical space. So all these dots will correspond to compounds. And from these slides, you see that if you compare natural products, for example, to compounds which were issued from combinatorial chemistry, you see that the space is much wider. And actually, this space matches quite well the one of drugs which are currently on the market. So that's why there was a strong interest to use natural products as a source of new drugs in general. And about 30% of all drugs that are currently on the market are still coming or are from natural product origin. And also these compounds also help to find new targets. Maybe they will not be translated into drugs directly, but they will inspire chemists to design novel active agents. And when you look at drug discovery, what you would like to have is pure and well-defined natural products, but the main challenge for phytochemists like us is that these compounds are indeed convoluted in a very complex matrix, which is a crude extract. And we develop technologies in order to profile this crude extract with a very high precision. And this helps us to either target the isolation of compounds of interest like this one, or also to standardize the extract to look, as this was mentioned by Michael, for bioactive compounds, active principle in phytopreparation, or looking also at potential toxic compounds. But for this, metabolomics is really needed. And having this deep knowledge on the metabolite composition gives us a lot of avenue and possibilities today, because if you master the metabolite composition of all these herbal extracts, you can first start to make some rationale about their use in traditional medicine. Through isolation and pharmacological assay, you can prove the activity of some of these active compounds. And today, with the development of the digital era, you start to be able to build connection between all these metabolite profile, their possible bioactivities, and this field is really blooming, and I will try to show you how we start to make this connection on a semantic web way to combine experimental data with prior knowledge. So very classically in our field, when we perform this type of study, we start with natural extracts. We perform what is called the bioactivity-guided fractionation, where we can follow activity of the extract constituents through fractionation with chromatography, obtain pure constituents, and do their characterization. So this path is pretty long, and because of this, most of the companies have refrained to do this natural product drug discovery, but they didn't really find very new chemical structure until now, and combinatorial aspects can help the development, but not really the discovery of new scaffolds. And in this process, what is important is to profile the extract to dereplicate known compounds and avoid their isolation. So that's how this technique starts to bloom in the field. And today, we are using especially LCMS for profiling the extract in a very comprehensive way. And this information are very useful because they give you a holistic view of the composition of extracts. And with bioinformatic methods, we can start to target the isolation of compound of interest. So it's really like finding a needle in a haystack with a powerful magnet, let's say. So to give you an idea of the complexity of the plant metabolome, we were interested, for example, to work on TCM drugs, which are extremely complex because that's a mix of many herbs. Here, 8 herbs, for example, for curing acute dermatitis problem here. And very strangely, if you profile the extract with quantitative methods like LC coupled with ELSD, you will see mainly one peak. And guess what is this peak? If I'm zooming, so when you start to zoom, you start to see other metabolites. But if you do, for example, an NMR experiment of the whole extract, that's the signal you will get. And actually, all this is related to sucrose. So that's, since this was a mix of roots of many medicinal herbs in China, of course, sugars are very important. And you see that quantitatively, there were mainly sugars. But there is hundreds of constituents where they are. They are sitting here. And if you profile now the extract with an LCMS method, you start to see them. And you can even go much more deeper. So that's a low-resolution chromatographic LCMS profile, but you can do it with very high-resolution aspects. And I know that Michael liked that because this is a way to deconvolute the extract. But this is just to show you how complex it can be. All these peaks correspond to different compounds, okay? And with LCMS, you can really do this at a very, very highly sensitive method. So that's what you will ingest when you take a TCM. So what are the toxic, what are the beneficial compounds? So that was just to show you on a technical viewpoint what we could get. And these profiles look like that if you have this coupling with LCMS. So basically, all these peaks correspond to compounds in the extract. And the methods allows you today to do this. I think I will switch it here because it's more easy like that. These methods allows you to detect not only the main compounds, but also very minor constituents at the same time with very untargeted methods in metabolomics. And basically, the information that you get, of course, is not directly the structure. What you will obtain is the molecular ion, from which you can retrieve the molecular formula. And automatically, you can retrieve what is called an MSMS spectra. And this is a fingerprint for the metabolites, and you can query this fingerprints against databases. The main issue with natural product chemistry is that these databases are not very extensive, and there is more than 300,000 natural products which have been published, but we have to find strategies in order to do this search with in silico methods. So basically, that's the type of information that we retrieve, so molecular formula and MSMS spectra. I will summarize this with these icons in the slides to come, and they can be looked at a first glance as a fingerprint of the molecule. Just to give you also a glance of the sensitivity, you have seen the resolution of LC. The mass spectrometry today is extremely sensitive and has a very high dynamic range. So it's a little bit like if you were having a high-resolution camera flying over the Amazonas, and your high-resolution camera allows you to see the leaf on the trees, for example, but also all the mushrooms below the trees, and even the insect grazing on the mushroom below the trees in the forest. Okay, so that's just to give you an idea on how sensitive can be the mass spec data. So sometimes you look at some metabolites and they are at this level. So you have always to have this in mind when you do this type of profiling. So in our group, we try now we work with several thousand of natural extracts, and we try to do this in-depth metabolite profiling in order to retrieve as much as possible chemical information to target the isolation of pure natural product, let's say in a targeted manner, and then assay only the very interesting molecules in bioassays. So for this, it's very difficult to interpret all the spectra. So we take the advantage of what is called molecular network. So molecular network is a method which allows you to make connection between metabolites in a given LC-MS profile. And this is done by looking at the similarity of the spectra of each of these molecules. And at the end, you will see that we are able to annotate a very large majority of all these metabolites. Have in mind that annotation by mass spec is just an approximation of the structure. It's not the full identification, but it gives to the chemist already a lot of information, and it helps also standardization of drugs and quality control. So basically for each of these dots, you will see that we can retrieve an LC-MS spectra for the molecular formula and its corresponding MSMS spectra. So if you look at a real extract, this is how it looks like here. This is a real profile of a given plant extract. Well, this was Arabidopsis thaliana in this case. And on this dimension, you have the mass spec, and here you have the chromatography. So every dot basically will correspond to a given metabolite. Sometimes you have more dots than metabolites, but to have a rough idea, that's how it looks like. So that's really the complexity of what we are looking at. And this is for a single plant. And for each of these dots, you will get the MSMS spectra. So you can get more than 1,000 annotation for a given plant extract. So if you look at TCM, it's even more complex, but that's just to show you how deep we can go into these data. So in order to annotate the metabolite rather correctly, it's very important to have a knowledge of what was published before, and for this we developed this Lotus initiative during COVID time, and my PhD and postdoc here, they were able to retrieve all publicly available chemical structure, and they make a link with their biological taxons. This is very important because every structure which has been published has to be linked to the source organism. And this taxonomical information helps you later on to do the correct annotation because, of course, we have to take into account the biosynthetic background. And this database is freely available on the web. It's here. And I think it's a very important resource for probably all those which are working on natural products because this helps you to make rational on the chemical composition of a given plant, for example. And you will see that we can link this also with pharmacological activity. So for example, in this Lotus database, we could query for any plant. So you can do a query like that. So the data are now in Wikidata, and we are just, because we could put them in Wikidata, which is a way also to keep them almost forever. But so our database now is querying Wikidata with the taxon data that we put in. And for example, you can ask, what are all the metabolites present in Arabidopsis thaliana? And this will display this type of graph where you see in which database you have the metabolites. And you can do this with, by writing this type of query, but I'm not very good at doing this. But today, you can ask ChatGPT, and you can say, write me a query to ask for retrieving all chemical structure from Arabidopsis thaliana, and it works. He wrote me this query, I run it, and I could retrieve 600 natural products reported for Arabidopsis thaliana. So, and this is also coming in the field. We will have this natural language interfaces to interrogate our experimental data. So, in Lotus, we could retrieve 700,000 pair and about 250,000 natural products. And this is the chemical space of Lotus. So every dot here will correspond to a metabolite. And through this way of displaying the compounds, they will be displayed by chemical similarity, you see. And this is a way to classify these natural products. And all of them will have the link to their biological organism. So all this information now is available to you on the web. And this is quite useful because for us, for the annotation aspects, we can now use all this chemical space because we have all the structure. And from this, we can calculate, and it's possible to do computation for this. This is starting not to work. Okay. We can calculate in silico MSMS spectra. So this is predicted fragmentation spectra of each of the molecules that you see in the chemical space. And now, with this, we can query our experimental data and do annotation. Of course, this type of query is not perfect because this is not experimental against experimental, but it's quite useful. So we have different ways. I will not go into details to compare this MSMS spectra against either experimental data here or against in silico spectra. So our strategy is mainly to build up this huge library of in silico spectra because also the condition may change from one instrument to the other, and systematically do this annotation with this strategy. So now we get the MSMS spectra for each of the molecule, and I'm going to show you how through molecular network we can make these links. So have in mind that every of these peaks will get an MSMS spectra. If the MSMS spectra is similar, that means that the structure are very likely to be similar. And by using this, we can build up molecular network. So a molecular network is a way to display any of the ions which have been detected, so the molecular weight ions, if you want, and in a kind of spectral space. And if a given molecular ion is given an MSMS spectra, and if another one in the same extract is given another MSMS spectra, if these two MSMS spectra are similar, and an algorithm can calculate this, then they will be displayed in the space very closely. And this will generate cluster. So you see, for example, here we have a cluster of molecules which are chemically similar because they exhibit similar spectra. Okay, so this is one structural type. And if you do this on an extract, you will have several structural types. So you will end up with a display like that, where you have different clusters. And every cluster will correspond to a given chemical structural type. So this is quite nice, because in an extract, you will already know what are all the major class of compounds. And you will know that you have analogs which can be connected by this way. So just to show you that it works, so we could generate, for example, here, the metabolite profile of about 300 U4BAC species. They are quite interesting, especially for anti-cancer activity, for example. And we could compute, can you start the video for me here, if you click on it? It's running, it's not running maybe, yes? I'm looking at the guy, but it's not running anyway. No worries, but what you would have to see here is that by calculation, you can start to build up clusters by MSMS comparison. and at the end, you will get this cluster for the whole data set. So after calculation, you will see all these different clusters, and this will help the chemist now to know where is a given structural type and annotate all the compounds. So this is for the 300 extracts. So we collected about 1 million spectra for this data set. And now I can show you what are the type of information that we have, for example, in one of these clusters. So for each of the dot will represent one molecule if you want. This is not completely true. There is many dots for one molecule, but have this in mind. Then for every dot, you have this MSMS spectra. We can do then the spectral similarity score for each of them. And now we have to annotate this compound. So in this case, for example, we had one standard, one pure compound, which was a 4-bol ester derivative. So this one was fully identified. We know that since they are connected in the cluster, that means that all the other dots should have similar structure. Okay, but what are these structures? So for this, we run our algorithm, which is comparing the MSMS spectra against in silico data, and the system will give us candidate structure for each of the dots. So we will have several candidate structure with a given score, and this is what we call the annotation. Okay, so this will annotate mostly all compounds, but of course with a given score. So we are never sure of the exact structure, but this is already quite a lot of information. In order to improve this score, we will use the information from Lotus. I show you the database, because here we have all the taxonomic data. And this will help us to sort out what are the correct candidate for a given structure, because we can take into account the biosynthetic pathway. So by taking into account the taxonomic distance of the organism, then we can re-rank the structure, the top 5, for example, and have a better confidence in the annotation. That's our strategy at the moment. So this is really improving the annotation score. Just to show you this, for example, on one of these clusters, for this ficus species, for example, in the first round, we were having this annotation for different flavonoids, but we were unsure, for example, of this one and this one, because such type of aglycone, for example, were not reported in ficus species, but when we applied this algorithm, we could correct this structure. They were still having the same mass, very similar MSMS pattern, but based on this, we could do the correct annotation. So this is really helping as a filtering method. So finally, if we apply all the tools that I show you, we were able to annotate quite a bit of this metabolite in this cluster. And they were matching with compounds reported in U4BAC. So we know, for example, that this cluster were really containing interesting 4-bol esters. I will show you later on what was the activity of this compound. And this was also proved because we could get known standard, which shows us that we were on the right track. The other information, which is very interesting, is that for every node, every mass here, we have the distribution of the mass across the different samples. And we can see, for example, that this one is shared between different extracts. And then it's very interesting because with this, you can make correlation, for example, with bioassay results. So just to show you also how it looks like on phytopharmaceuticals, you probably all know ginkgo biloba. I'm trying to use it a little bit to improve my cognition. I don't know if it works really, but that's anyway what I'm ingesting, okay? And if you look at the metabolite composition of ginkgo biloba and you apply our methods, if you look, for example, at all the peaks which are detected at 5% threshold, you will get about 84 annotation. And that's mainly corresponding to what was published on ginkgo biloba. But if you go down in the forest to see the insects grazing on the mushrooms, you see that we end up with about more than 1,000 annotation for the same ginkgo biloba extract. And this is real compound. This is not noise. I mean, the metabolites are there. So I know that Michael is always asking me, did you detect some pesticides? So maybe they can be in this very low background. And that's anyway things which are taken by people. So the main question is what threshold is meaningful. That's really an issue for us. But that's just to show you that we can go at these two different levels. And I had a PhD who ended up last year who actually get an award just yesterday in Dublin. We were having a conference over there. And Adriano developed a method which allows us now in metabolomics to distinct what is the major metabolome and what is the minor metabolome. Because in mass spec, it's extremely sensitive. You will detect a huge amount of things. So we are trying now to use detectors, which are generic detectors, which give a kind of semi-quantitative response, which is not the case with mass spec. And we use these methods to filter the DMS data in order to look at the major metabolite if we want, or only at the minor one. But that's an information which is very important. And he developed a very complex algorithm which allows you to deconvolute every HPLC peak to understand what were the ions behind. I will not go into details. But basically, when you profile a plant with this, on the top trace here, you have this semi-quantitative trace, which already show quite a lot of constituents. And on the bottom trace, you have the MS part. And by applying this filtering, we could now go down from thousands of features to about a few tenths of main constituents and a few hundreds of minor ones. So this is something which starts to be workable and well filtered for annotation purposes. So we are really at the beginning of this part. But I think this type of information between major and minor metabolites are very important, also from a toxicological and pharmacological viewpoint. So now, I show you only chemistry. I'm a chemist, so that's why. But I still have to show you that we can perform some bioassay on all these extracts. So at the moment, we are trying to also screen the extract with different assays in collaboration. And we try to make connection between this molecular network and bioactivity. And you will see that this allows us to prioritize interesting compounds. So, for example, if I took back the example of this U4BAC set that I showed you before, all these extracts were also assayed. They were assayed, so that you remember, that was our molecular network. And now we have all this metabolite profiling data. And these plants were assayed for Wnt signaling pathway in collaboration with Vladimir Katanaev. And you are more specialized than myself on Wnt signaling, but it's quite interesting for carcinogenesis, and we can find different compounds which are modulating this pathway. And in our case, about 20 extracts showed a very good inhibitory activities on Wnt. And we were trying now to make a connection between these 20 extracts and the 300 that we were profiling. So for this, we took again our molecular network, which is here on the right, and we color code all the cluster according to the bioactivity we get at the extract level. So all the ones which were in blue were inactive, and in red they were active. So you see, mainly on the whole network, it's mainly blue. But then we could really look at the level of every cluster if we were having red color or orange color. So we add metadata to every of these dots. And this allows us to finally select 21 clusters among 2,300 for the whole data set, which were clearly linked with bioactivity. And I will just show you one of these clusters, how we could deconvolute this cluster. So you see there was several metabolites which were linked to bioactivity here. And I can just compare on the same plant clusters which were inactive. So the inactive cluster were showing very different type of structure, but the active one were indeed from the 4-ball ester type, which was quite correct also based on prior knowledge. But of course, with mass spec data, we didn't know exactly the structure. In order to find the real correct structure, even if we have annotation, you need to isolate and go for NMR. And that's the work we do also in the lab. And what we develop on our side is that we have this very precise metabolite profiling data, and we are able now to translate this at the analytical HPLC scale to find out where are the compounds of interest based on mass spec. And now we can go to a semi-preparative scale to follow the same compound and isolate them for full de novo identification. So we don't do any bioassay guided fractionation. We can just do a cherry picking of the one which are of interest. And we can do this at small scale. Urs knows this guy, Emerson. So this is the Pope of isolation in my lab. So Emerson works at different scale, you see. HPLC scale, flash chromatography, and even MPLC. Just to show you that if you need to isolate hundreds of milligrams of compounds, you need to go on this part. In India, probably I would need this to get standards, for example. So basically, with this approach, now we are able, after isolation, to get the correct structure of these compounds, and through, of course, interpretation of the NMR data. And very interestingly, in this case, we pop up with this 4-ball ester. And if I look at the structure of this guy here, actually, the structure of this 4-ball ester was very similar to tigilanol tiglat, EBC46, and this we found it in a euphorbacea plant from New Caledonia, and this one was found in an euphorbacea from northern Queensland. But the one on the right is known to be an activator of PKC, and ours was a potent Wnt inhibitor. And the one on the right actually underwent phase 2 clinical trials for you see, a given type of cancer here. So these diterpenes are very promising as an anti-cancer agent. So that was a proof of concept that we could really cherry-pick some compounds, and we end up with very strongly bioactive compounds. So I show you this for 300 extract, but we can work at a larger scale. This was not complex enough. I have to go a little bit further. So we have the chance to have the collection of 15,000 plant extracts from the Pierre Fabre collection. And at the moment, they are not doing any more drug discovery on this, but we can profile part of this, about 10% of the whole collection, with the method I show you. And this gives us access to the chemical space, or the virtual chemical space of all these extracts. So now we try to make some queries, but you see, it starts to be extremely difficult. This is the molecular network of about 1,600 extracts, and it goes further. So it becomes impossible to visualize the data. And our idea is to convert this into a virtual chemical space for querying and using artificial intelligence algorithm to help us to do decision here. So how to do this? Well, this is really a complex metabolomic set, and you know that people working in metabolomics like to find needles. So I'm not finding needles myself, but I have a very nice crew of people who are developing tools to look at the needles in this very complex haystack. So the limitation that we were having is that with molecular network that I showed you before, we cannot visualize the data anymore. Because for any of the dots in any of the cluster, we have many annotations. So how to find a way to work with this? And we are reproducing this metabolite profile 1,600 times, and we have still 15,000 extracts to look at. So it's quite a big work. So how to deal with this? Actually, and this is very new, we try now to convert our experimental data into a semantic web format. And actually, this is how the web is organized. If you look at Wikidata, this is semantic web data. And the idea, and this is a few groups are starting to do this, is to convert your own experimental data in the same format, because then you can do expert queries. So the idea here was to deconvolute any of these extracts. You remember the dots correspond to molecular ions. Every dot will have an MS, an MS-MS. Every dot will have different structure, is spike code. They will belong to a given LCMS spectra. And on the top of that, and this is most of interest for you, there is a biological activity. Okay? So now, you can build up what is called a graph, a knowledge graph, and it uses this RDF format. So this is how the semantic web is organized. Just to show you an example, you probably know this lady. Okay? And so actually, Meghan Markle is the subject, and Prince Harry is the object. Don't tell Prince Harry that he's the object of Meghan Markle, but they are linked through a predicate. That's how it works on Google, okay, on the semantic web. And there is also information. For example, Meghan Markle is played by Rachel Zane in Suits. I never follow Suits, but it seems that is the case. So now if you do a query in Google and you say, who is the husband of the actress playing Rachel Zane in Suits, the answer will be Meghan Markle, but you see that there will be some relation with Harry, but also with his previous husband, Trevor Ingleston. So that's just to show you how you can query the web, and you can query the data the same way. That was mainly my message here. Okay, so forget the very complex computational aspects. We develop many tools now which are putting together structural annotation, also prediction of spectra, linked to different databases, and we can query our data in this way. Just to show you how they look like, this is now all the different features, the LCMS data, their annotation. Every annotation has a spectra. So that's how the graph information are. And I will not go into details, but if we deconvolute completely all this information, every object is linked to the next one as a triplicate. And if you do this on these 1,600 extracts, and especially if you link this, and I think this is coming now, with bioactivity, because every of the plates containing the extract has a given bioactivity, the information is there. You need to query and see how you can make your connection. So we have done this, and now we have about 40 million triples in the database. And so everything is connected, okay? What is interesting for us is that now you can connect bioactivity and extract composition. So a given extract has a given composition, and you can start to make query to see if statistically there is some of the compounds which can explain the bioactivity of the extract. And it's quite powerful because the data are organizing in this semantic web format, and the web is organized the same way. So you can start to make links between the platform knowledge with the web knowledge. So for example, we are starting, this is really new on our side, we are starting, this one is not working anymore, to make a link with ChemBL, for example, where a lot of pharmacological information are embedded. But through the structure, we can start to make these links, and we can generate hypotheses, okay? So we have been doing this, we start to do this on this data set, and now the idea is we cannot look at all the metabolites, but all the metabolites explain one extract. And now every dot that you see here is the whole spectral information of one extract. So every dot is one extract, 1,600 extracts. And we use algorithm to see if extracts can cluster together based on chemical composition. So for example, here with this visualization algorithm, you see that we have two clusters here as well. And these clusters, for example, here highlighted in green, were the active extract of the same data set for antiparasitic activity. So we were having 7 extracts out of the 1,600 which were active in this case. And they cluster together. So now we have to know why they are clustering. Is there any features which are common to this extract which can explain the bioactivity? Okay? And actually, this was the set of plants which were highlighted this way. And taxonomically, they are not related. So there should be some chemical elements which are common. And we can do queries. So we can now start to write Sparkle queries, like on the web, on our data. And for example, here, we look at the rate of annotation. So we ask to our graph DB what were the chemicals which were more frequently annotated in the active extract compared to the whole data set. And we end up by looking at the class of rotenoids here. So there was much more rotenoids in this extract which were annotated than the whole data set. So now, with this, we can make the assumption that indeed these 7 extracts have rotenoids inside. And indeed, we could, by looking back at the data on the mass spec side, we could indeed find traces of rotenoids in all of them, mainly these 2. And actually, in this case, we could even order these 2 to prove that they were inside. So, and these are known also to be rather toxic molecules. But interestingly, at least in the assay that we perform in Basel, they had a very interesting selectivity index, which was a bit puzzling, but at least this could have been toxic molecules. But you see, this is just a proof of principle. We could get this just through the statistic information, and straight away we highlight this compound. At the moment, we could isolate all these ones, and we are checking if there is really a selectivity aspect. So that's the beauty of plants. You can get a whole series of compounds, and you can check if there is any structural activity relationship, at least in this assay. So for us, this was a proof of concept, and now I will finish with this. You remember, this was the chemical space of all knowledge data from Lotus. Okay, that's what I started with. And now, with the metabolite profiling method I show you, we start to be able to annotate. And on the 1,000 seed extract from Pierre Fabre, which were really biodiverse, in blue, you see all the annotation that we could get. So you see that already with this 1,600 plant extract, we cover quite a large part of the chemical space, which is known to date. There is parts, for example, here, which are not covered at all. Why? Well, this is mainly coming from the extract, because Pierre Fabre was doing extract which were targeting molecules in a given log P range in order to be druggable. But, for example, here, you can have, for example, glycosides, which will not be considered as druggable from this rule. So that's explaining why we have some holes. But this is showing also that even with a small set of plants, we can start to have a rather high chemical coverage. So now we try to use all our annotation as a virtual chemical space, and we are really at the beginning of using this data for doing artificial intelligence-guided isolation. The very final things I would like to show you is how we can link this data with ChemBL. So, for example, this is what you see here in blue is all the annotation that we retrieve from Pierre Fabre. And in yellow, what you see is all the structure that we have in ChemBL. So, so we were looking at IRA in this plot where we were having both dots which were in ChemBL and the relationship with metabolites in our data set. So here on this branch, you see these two compounds, and that's the structure of these compounds. And they were interesting for us because they were reported to have a rather interesting IC50 value in an assay against Leishmania donovani, which were also screened in Basel in collaboration. And we were interested to know what were the natural products related to this one, which were in our data set. So we found the ions. So if we know the ions, we can find in which plant they were belonging to, and based on the mass and the retention time. So finally, we identified two plants where these compounds were reported, or analogs, and we could target the isolation of these compounds. So we could retrieve finally, about 8 or 9 of these terpene analogs to those which were bioactive in Campbell. And we isolate this with the targeted isolation that I showed you before. And we could retrieve some interesting activity for some of the derivatives, just to show you that this gives us also a possibility to explore analogs. So either we explore analogs or we explore parts which are completely uncovered. That's a little bit the idea. So with this, I hope that I could convince you that we are at a point where with metabolomics, we start to be able to do very detailed metabolite profiling of the extract, bridge this with taxonomical information, create virtual chemical space for queries, and use also semantic web data to make a relationship. And I think it's important not only for drug discovery, but probably if we evolve with this method, we should maybe start to have possibilities to make relationship with phytotherapeutical herbals and maybe understand a little bit better, or at least have clues on how this is working. So, of course, very importantly, this has to be linked to bioassays. And hopefully, at the end, this will really be useful to help the targeting of pure natural product or the chemical composition assessments of herbals. So with this, I would like to acknowledge all the people in my group. This is a rather old picture, but you see they were very dynamic. So, and we showcased ourselves recently on a chemia paper in the form of a molecular network. So that's all those which were mainly involved in this, the development of the tools I show you. We are in Geneva, near from the Geneva fountain. Caroline is with us, so sometimes she's in the same building as we are. And we have the chance to have this NMR and mass spec facility, so we are very open for collaboration with all of you. With this, I would like to end up, but before just ending, I have to advertise you also a little bit, because last week, no, not last week, but yesterday I was in Dublin at a GA meeting. So the GA is the Society for Medicinal Plant Research and for Medicinal Plant and Natural Product Research. I think the activity of GA are very resembling to what we are doing here at IUFAR. IUFAR is probably much more pharmacologically and toxicology oriented. We are more chemically oriented at GA, but we were discussing also with some of you last night, and it would be interesting to maybe bridge some of the activities of our societies. GA is very open for this, and I would just like to advertise you that we will have next year in Krakow our joint meeting, which is a very big meeting for all natural product chemists, and it would be interesting to have also more pharmacological talk and toxicological talk related to natural products. So please be welcome to come in Krakow, and thank you very much for your attention.
Absolutely marvellous. When I became Secretary General in 2014 of IUFAR, one of the key things that I wanted to push was evidence-based science for natural products and their identification. So Jean-Luc was therefore one of the key factors of that, and you can see how his work's developed, which I find extraordinary. So, questions, please. Dr. Bikash?
Yes.
When you look at metabolite, and we look at how many are active metabolite. So when we take the product into regulatory pathway, so many regulators, they said that you have to identify at least 4 active ingredients and some metabolite. And can you make it a point, like you said that you look at the chemistry part. So when you go for further, so if the active metabolite is the pharmacologically active, and at the same time, if you have a mechanism like you said about artificial intelligence, can you predict how many of them can develop an ADR also at the same time?
Can develop, sorry?
Adverse drug reaction or adverse event.
Yeah, so this is, of course, extremely challenging, but indeed, that could be one prospect. So indeed, for the regulators, I think, well, depending on the country, they are asking for at least being able to characterize a few metabolites, usually a few. It's like 4 in India. In China, I think they are asking 1 active principle per plant if they do a formulation. And you see that if we go really in depth, we can annotate hundreds. So we don't have to show this too much to the regulator as well, because they might ask for more. but the point is that, so on one side first, we have to, I think this, I show you very quickly this minor and major metabolite classification for the industry, for standardization. I think you can use our tool with this filter first to look at the major metabolite, because anyway, not maybe for toxicology, but at least for pharmacological effect, they are the more likely to have an effect. So we get the structure indeed. So from this, if you identify the active principle, this has anyway to go through isolation and some bioassays. And you know that in herbals, we don't have always the active principle. So if we don't have it, you can use it as a standard, as a marker, taxonomic marker for the plants. But indeed, your question was if you have the active principle through maybe virtual metabolization, can we predict the toxic aspect? I think starting from the structure, because what we are just trying to deliver is structure. If there is any program which allows you, for example, first to predict metabolization, and they exist. So you can predict all possible metabolites for a given structure, phase 1, phase 2. And from this, you can probably, if any data would exist somewhere on the web on these metabolites, or maybe analogs, you can start to make links. But we didn't do it, but I think, yeah, potentially it's possible, provided that the structure is correct. Yeah. You can, yeah.
It makes a lot of difference when you go to the clinical part.
Yes.
Like if you look at a giant in India, like Himalaya, natural remedies, they usually collect in a particular time, like in the rainy season, summer season, and that activity is, it is huge differences there when you go for a clinical efficacy. So is it database when you're going to create the data like you had done, is it going to reflect the diurnal variation or in terms of collection for the rainy season or other season, and where you can see that a lot of differences there in terms of efficacy in a clinical setting?
Yeah, no, that's a very good point. So what I show you were mainly static data, so metabolite profiling at a given moment. But indeed, a plant is not a plant. That means that all the biochemistry reacts to the environment, and it's well known that moment of collection will change the metabolite profile. So you can use the same method to profile this at different time of the year, for example, and this is done in metabolomics, and observe the changes. And indeed, in future, if hopefully everybody can share metabolome data, this information should be provided as metadata to know if this metabolite profiling was done in which region, at which timing, and this will start to give clues. For example, on the ficus data, we were with the Malaysia. I just show you one cluster with the flavonoids. This was the purpose of the study. So we analyzed about 80 of these plants at different timing, different location.
Okay. Two very quick questions, and then go on hard, but please quick.
Okay, Tanya Garkovic, NCI, and I have a practical question. I'm also a chemist. My question is about time. So you've shown lots of beautiful UPLC traces going from 30 to up to 120 minutes. I'm assuming you're also hitting multiple collision energies in your MSMS spectra. So how long did it take, not only to generate the 1,600 plant extracts, but also to process them? Because I'm assuming your data files are huge.
Mm-hmm. Yes. So for the profiling of 1,600, we went to shorter gradient than the one I show you. So usually we profile about 100 plants over a weekend. We do 8-9 minutes gradients. So the acquisition is about 100 plants in 2 days for the acquisition. Then the processing is indeed a major issue. At the moment, for the 1,600, to compute all the annotation, it took us about 3 weeks of CPU time. But now we are mainly privileging this filtering that I show you with semi-quantitative data. And the idea is to have a completely fully annotated report of about a few tenths of main metabolite, and this can be done in a few seconds. So, but this is just there. We are published. The last one is not yet published. We submitted. So this will give a rationale for this part. But we can discuss about that.
Okay. Go on quickly.
Okay, so thank you for your presentation and good work. As you talk about the molecule network, I think it's very important for us to understand the compound and how to analyze the bioactivities for the extract based on the network, molecule-based network. This is one question. Another is, as you talk about the mine dates. But when we mine the dates, we want to find some active compound or active extract or fractions. The question is which one? The pure compound, a single one, or with the complex of compound is our goal.
Okay, so
thank you.
So yes, so with the network to find out the active. At the moment, we are doing this connection with many extracts. So we use the high number of extracts to make our correlation. And you have seen that we can find out that a given ion can be found in many extracts. So we use statistics based on a high number. And based on this, we can say, okay, most probably, since this ion is always popping up in different active extracts, it should be the one. So that's how we could say it might be the one. But of course, in sometimes you need to have fraction data, and at the end you need to have the pure compound. So that was for the first question. For the second one, indeed, when working with herbals, we all know that it's very tricky to find only one active principle explaining the whole clinical effect of the herb. You have seen with TCM, for example, there is many compounds. So at the moment, we can only offer a way to identify and assess the activity of single compound. But of course, synergistic effect has to be taken into account. Metabolization have to be taken into account. So there is still a big gap between chemical composition and clinical efficacy. But by building this knowledge, maybe, and looking also at results in general, maybe finally we could get this type of links to be done.
Thank you. Following Jean-Luc's wonderful presentation on chemistry and phytochemistry, I will do my best, okay? So my name is Valerie Schini-Kerth from Strasbourg University, and we are interested in natural products and the health effects. And why are we so interested? Because cardiovascular disease, as you know, is still the major leading cause of death. It's increasing globally. You can see that the number of... oh, is it working? Yes. This one is over, I think. Yeah, that would be good. Okay, as you can see on the left-hand side, the number of deaths is still on rising lately, as well as the prevalence and the years lived with disability. So there's still a big challenge how to protect our cardiovascular system. And this increase in cardiovascular disease is mostly linked to the aging of our population. You can see from the 1950 to the estimated what's happening in 2050, we have more and more people older than 80 years of age. And of course, age is an increase, cardiovascular risk. You can see on the green light, with increasing age, there is a lifetime risk associated. And this is further increased if you have one risk factor which is not optimal, even further with one elevated risk factor, even with one major risk factor. And of course, if you accumulate risk factors, you can see that cardiovascular events may appear even much earlier. And those cardiovascular risks are mostly linked to, I can see, yes, those cardiovascular risks. You can see here on the right-hand side, of course, smoking is a major cardiovascular risk, increased body mass index, physical inactivity, an inappropriate diet, and of course, metabolic diseases like increased levels of cholesterol, LDL cholesterol, increased systolic blood pressure, or diabetes. And the risk associated with cardiovascular disease is mostly the origin is ischemic heart disease and stroke. You can see that ischemic heart disease accounts for about more than 49% of deaths, followed by ischemic stroke. Now, what is the underlying mechanism of those ischemic heart disease? First of all, which are the areas they are affecting? Mostly the heart circulation, the coronary circulation, leading coronary heart disease, heart failure, but also brain circulation with stroke and peripheral artery disease. Those are the blood vessels which are at risk. And the underlying mechanism is mostly the development of arteriothrombotic events. This actually starts very early on in your life. You can see, starting from the 4th decade, we can see appearance of early lesion of atherosclerosis. Of course, no clinical impact, which will further progress and become an atherosclerotic plaque. And this plaque the problem can be, there can be a lesion or a rupture which leads to an atherothrombotic events, and of course, if it happens in the coronary circulation, can lead to myocardial infarction. And when we look carefully at the development of these atherosclerotic plaques, we can see that very early on, what we can see as an alteration is actually the appearance of endothelial dysfunction. This is one of the early events we can detect even at the stage of atherosclerotic lesion. And this endothelial dysfunction is linked to an impaired formation or an increased degradation of nitric oxide, the major vasoprotective factor, increased oxidative stress, monocyte adhesion, lipid accumulation. So then, of course, we need, if there are approaches to protect the endothelial dysfunction, the initiation and development, this would be an interesting approach to prevent the development of cardiovascular disease. Now, lately, it has been published here the top risk factor related to overall disease burden And you can see the top one is actually the dietary risk. An unhealthy diet is by far the most important risk factor, followed by tobacco smoking, high blood pressure, body mass index, physical inactivity, and then the diabetes and hypercholesterolemia. And very recently, the American Heart Association, but also the European Society of Cardiology, has very important messages for the public regarding health. You can see that the cardiovascular burden is attributable to modifiable risk factors. Here we have the 8 modifiable risk factors indicated: the diet, physical activity, blood glucose level, increased weight, hypercholesterolemia, high blood pressure, smoking, and a relaxing sleep. And when we look carefully on the right-hand side, well, you can see the prevalence estimates of poor, intermediate, and ideal cardiovascular health. You can see that among the 7 modified cardiovascular risk factors, the one which comes out the worst is the diet, a healthy diet. And you can see that those are data from the USA. Regarding a healthy diet, we have less than 0.2% of the population has a healthy diet. Less than 20% have an intermediate diet, and more than 80% have a very poor diet. Now, how can such a diet impact the cardiovascular system? And what can we do? You can see here there is now more and more evidence indicating that actually our diet is very important for protecting our cardiovascular system. Here's published the first primary prevention study regarding the impact of Mediterranean diet on the cardiovascular system. They enrolled 7,447 persons. They were not persons with a high cardiovascular risk who were free of cardiovascular disease when they were included in the study. And they were either on a controlled diet, there was people from Spain on a controlled diet, or a Mediterranean type diet with daily intake of nuts or extra virgin olive oil. And you can see over the 5 years of the study, there was a very huge benefit with a relative risk reduction for approximately 30%. This was a primary prevention study. And here they also very recently on the same topic published in a secondary prevention study. You can compare the Mediterranean diet to a low-fat diet in preventing major cardiovascular events. It was the CardioPrev study in 1,002 patients with established coronary heart disease. And the Mediterranean diet was compared to a low-fat diet, and again, the Mediterranean diet did much better with a 26% reduction in the risk, in the relative risk. We also have some interesting data here from a study from China, where they looked at the intake of red food consumption and major cardiovascular disease in China. It's a very big population, more than 500 adults, and they look the development of cardiovascular death or major coronary events with portions of intake of fresh food. And you can see a negative relationship. The more food intake the people have, the reduced risk they have for cardiovascular death and major events. And what is interesting, 30 portions per month, which is 1 portion of food per day, was actually associated, you can see here, with 37% reduction in the risk of cardiovascular death and a 30% risk reduction in major coronary events. So really very strong effect. Now, how can we explain this impact of the diet on the cardiovascular system? And we know since Professor Fürsch's discovery of EDRF, which is nitric oxide produced from endothelial anosynthase, the major vasoprotective factor in the cardiovascular system, which can be modulated by a lot of different local factors, but also the most important one is shear stress activating endothelial cells to produce nitric oxide in a continuous manner over 24 hours, and this NO is important for vascular production, but also a strong inhibitor of platelet and monocyte adhesion and activation. And we know now since more than 20 years, when the studies have started, that actually there are nutrition-derived activators. We can exactly do the same thing, activate endothelial cells to produce nitric oxide. We have cocoa polyphenols, red wine polyphenols, berries, and green tea polyphenols. I would like to share with you an overview of some of the studies we contributed. And you can see here very early on in 2000, we studied the red wine polyphenol extract. Only the polyphenols were isolated from red wine from the French Ministry of Agriculture. And the red wine polyphenols in porcine coronary arteries contracted either with endothelium intact artery caused full relaxation. We removed the endothelium, absolutely no effect anymore. So we have a strict endothelium-dependent relaxation. And what is interesting to see, here we have the concentration response curve. You can see 10 microgram per milliliter of the extract. We have nearly 100% relaxation. And just to indicate you that 3 microgram per milliliter of polyphenols have been detected in plasma of a subject after drinking 1 glass of red wine. When we look at the mechanism underlying this relaxation, we could see that nitroargine, the inhibitor of endothelial anosynthase, significantly reduced the relaxation. It was also inhibited by the 2 toxins here, which inhibit the endothelium-dependent hyperpolarization component, and the combination of 2 was associated with a full inhibition of the relaxation, indicating that this polyphenol, when polyphenol-induced relaxation, is mediated by nitric oxide and endothelium-dependent hyperpolarization. As a pharmacologist, of course, we do not want to motivate people to drink red wine. We have, of course, the ethanol component, which has a deleterious impact. So we were looking for different sources of polyphenols being able to have a beneficial impact on the cardiovascular system. And for this, we worked with the food industry. We were able to get here 13 different food, either as a juice or as a puree, which are used by the food industry in Europe, and we tested the activity of those compounds in the coronary artery system to do a kind of screening. Here in each graph you have in red, it's the same curve, which is the Concord grape juice, which is a potent activator of the endothelial formation of nitric oxide, and we were able to identify 5 products here which are more active than the grape juice. Here we have 3 products less active, and here we have products with very little activity. And this was very surprising for us because all the products have a high level of polyphenols, more than 1.7 gram per liter. So it's not, if you have a product with a high level of polyphenols, that you will have biological activity. You really need to identify what are the compounds, and as Jean-Luc said, we need new techniques to identify which structure is responsible for this activity. And again, the ones with the highest activity, you can see here on the left, is the aronia berry, the blackcurrant berry, blueberries, cranberry, and lingonberry. Those products are products really of major interest for the cardiovascular system. And we also looked, based on our studies, what is actually on the shelf of the supermarket. What kind of products do we buy as consumers? And here we compare 6 different fruit juices, which are blackcurrant juices, which are you can buy in the supermarket in France and on a neighbor city in Kiel, Germany, just crossing the Rhine River. And you can see actually that what we observed is actually the biological activity to induce endothelium-dependent relaxation was actually quite heterogeneous. Some food juices had very strong activity, and some had a very poor activity. And this is, first of all, important because it means that as a consumer, what we buy, we have no idea about the intrinsic quality of those juices. And with Joël Pansmail and our colleagues from the University of Liège, we looked at the antioxidant activity of those juices and the biological activity to see whether or not the antioxidant properties is of importance. And actually what we were able to see is actually the antioxidant activity is not associated with the biological activity. However, when we analyze the anthocyanin content, which gives the color to the blackcurrant juice, you can see that the total anthocyanin and also the individual anthocyanins were highly correlated to the activity, indicating that anthocyanins are involved in developing the biological activity of the blackcurrant juice. We also were interested to identify, as pharmacologists, what are the underlying mechanisms leading to endothelial cell activation. And here we stumbled in some surprising data at the first time. You can see here on the left-hand side, so here we do the experiments in presence of indomethacin and caryoptotoxin and apamine, just to focus on the nitric oxide-mediated relaxation. So this relaxation to Concord grape juice is entirely due to nitric oxide. And you can see when we based on the experiments, we tested whether or not there was a pro-oxidant response in endothelial cells, triggering activation of endothelial endosynthes. And you can see that the SOD membrane permanent analog, MNTMPYP, had a very strong inhibitory effect, whereas extracellular SOD, which is not able to cross membranes, was inactive. We also looked whether or not hydrogen peroxide was involved, and you can see that PEG catalase, able to cross membranes, also very strongly inhibited relaxation, whereas extracellular catalase was inactive, indicating that somehow we have an intracellular pro-oxidant response in endothelial cells, triggering the cascade leading to the activation of endothelial endosynthes. And this cascade was actually involving the activation of SARC. You can see the PP2 inhibitor significantly inhibited, and also PI3 kinase was inhibited. Workmannin, a classic inhibitor, is inhibiting the relaxation significantly. And indeed, when we look using Western blot analysis on cultured coronary artery endothelial cells now, when we expose to the Concord grape juice, you can see within minutes here, you can see an increased phosphorylation first of SARC, which will trigger the phosphorylation of AKT, and AKT is well known to activate endothelial endosynthes by phosphorylating serine 1177, which is an activator site. And you can see very strong phosphorylation up to 30 minutes. And this phosphorylation, this activation of this cascade, SARC, AKT, eNOS, in response to Concord grape juice, is highly sensitive to inhibitors of oxidative stress. Here you see PEG catalase, PEG SOD, and MNTMPYP, the membrane permanent analog of SOD, indicating that superoxide ions and hydrogen peroxide are involved in the activation of this cascade. And we were of course, interested. If you want to protect the cardiovascular system, it's not the bolus of NO which will protect in the long term. You need a little bit more NO formation, like with shear stress, in a sustained manner. So we looked here, activation of endothelial cells. This is the Aronia juice, and you can see up to 24 hours we looked here. And you can see phosphorylation of AKT is maximum 2 hours, then decreases slightly. But phosphorylation of eNOS is observed for 24 hours. It's a very long-term event. And again, we still do not know exactly how can we explain such a sustained effect. And when we looked at the control level of eNOS protein level, the non-phosphorylated one, what we were able to see was actually a time-dependent increase in the level of eNOS protein. And that's why we were also interested to see whether or not natural products may stimulate the expression of, upregulate expression of eNOS. And you can see on the right-hand side, I should not forget, bradykinin is a physiological agonist, and you can see that the phosphorylation of eNOS is very transient. After 30 minutes, there is absolutely no activation of eNOS anymore. Whereas with a polyphenol, an active polyphenol product, you can see, we see up to 24 hours activation of endothelial endosynthes. Very interesting. And so we went further to assess what is the effect of polyphenols on expression of endothelial endosynthes. And you can see data with purple grape juice. You can see with increase in time, there is an increased level of eNOS mRNA. We have an increased protein level here at 8 hours, at 24 hours, and this is associated with an increased formation of nitric oxide here, indicate after 24 hours. So indeed, we have a long-term effect, not only short-term effect stimulating NO formation, but also a sustained effect associated with an upregulation of eNOS and continuous formation of nitric oxide. This is exactly what we are looking for to protect the cardiovascular system. And when we looked at what is the underlying mechanism accounting for the upregulation of eNOS expression, you can see here the increased eNOS mRNA level in response to Concord grape juice is also significantly reduced by using the SOD and catalase membrane permanent analogs. And we could demonstrate that indeed Concord grape juice increases the formation of ROS, here assessed by dihydroetidium, and this is prevented by the PEG catalase and the SOD mimetic. And you can see here we looked about more precisely what is the mechanism involved in the upregulation of eNOS expression. And we looked at the FoxO transcription factor, which are actually negative regulators of eNOS expression. And we could see that the concurrent grape juice causes phosphorylation of the two FoxO transcription factors. This is a redox sensitive event. And when FoxO 1 and 3 are phosphorylated, they are no longer associated with the eNOS promoter, which they can no longer inhibit eNOS expression, which of course will contribute to eNOS expression. So based on this first part of the study, we have now identified that anthocyanin-rich products are of interest to protect the cardiovascular system. And they activate endothelial cells via a pro-oxidant mechanism in endothelial cells, leading to the activation of the PI3 kinase AGT pathway, phosphorylating eNOS, and stimulating eNOS production formation in a sustained manner. And also, long-term effects leads to upregulation of eNOS expression and more eNOS protein, of course, sustained formation of nitric oxide, which means that they are interesting products to protect the cardiovascular system. Now, when we look carefully in the cardiovascular system, atherosclerosis will develop not everywhere in the cardiovascular system. It develops in the coronary circulation, in the carotid bifurcation, femoral bifurcation, at the renal level. And when you look carefully in the coronary circulation here in the schematic, you can see in black, this is where actually the development of atherosclerotic plaques is happening. And you can see those are bifurcations in the main coronary arteries. Now, why are those bifurcations at such a high, at the target? And this can be explained to the fact that where you have bifurcations or curvature, you have disturbed flow, low levels of shear stress, which means endothelial cells are not activated in an appropriate manner. That means they will not produce enough nitric oxide to protect the arterial wall. And you can see this compared to, for example, the aorta, which is, of course, a tube, and where you have a steady laminar flow, high levels of shear stress, high levels of NO formation. And you can see this very easily when you look what's happening here in the aortic arch. You can see at the inner curvature, you can see that there is expression of vascular cell adhesion molecule. This is an activator of endothelial cells, whereas at the outer part of the ascending arch, you can see absolutely no expression of VCAM. And when we look very carefully here, this is the aortic arch, which is open. You can see that the inner curvature presents senescence. Senescence-associated beta-galactosidase, the blue staining, is in the inner part of the curvature, but the outer part is protected. And this is due to the fact that we have a deficit in NO formation. Less NO in the inner curvature leads to activation of endothelial cells, expression, for example, of VCAM. Now, senescence is a very important event because there are now more and more data indicating that actually premature senescence of endothelial cells is associated with several cardiovascular risk factors. It is induced in endothelial cells by oxidative stress, high levels, but also by activators involved in cardiovascular risk, like angiotensin II, oxidized LDL, high glucose, inflammatory cytokines. And this causes a major change in the biochemical activity of endothelial cells. They show senescence-associated beta-galactosidase activity, expression of markers of senescence, inhibitors of cell cycle like P16, P53, P21, excessive formation of ROS, but not a low level, a high level of ROS, and of course, eNOS will be downregulated, NO will be decreased, leading to endothelial dysfunction. Now, there are very interesting data published in 2008 in Circulation Research indicating that the induction of senescence leads to endothelial cell dysfunction. It's a very early event. And what this group did, they actually overexpressed the P53, which is a marker of senescence inhibiting cell cycle, only in endothelial cells. You can see here beautiful endothelial cells, which is expressing P53, The control vessel has no overexpression of P53. And what is the consequence at the functional level? You can see that expression of P53, specifically in endothelium, is associated with a significant impairment of the endothelium-dependent relaxation. And also, the formation of nitric oxide is significantly decreased, indicating that senescence leads into endothelial dysfunction. And this is why we were interested to see what is the effect of active polyphenols on endothelial senescence. For this, we prepared a blackcurrant extract so that we work only on polyphenols. We're using chromatographic column. And what was interesting to see, and that's why we shifted to prepare blackcurrant extract, because in blackcurrant juice, we have anthocyanins, but there are 4 major ones. And the 4 major ones are mostly conjugates of delphinidine to glucose or rutinoside to different sugars, and also cyanidine conjugated either to glucose or to rutinoside. And we were able to test the different compounds. First, to show you that the blackcurrant extract causes endothelium-dependent relaxation. It's really a strict endothelium-dependent relaxation. You get rid of the endothelium, absolutely no effect. And again, highly effective. And you can see here on the right-hand side the formation of nitric oxide assessed by this fluorescent probe, DAF-FN. You can see that the formation of NO is actually very similar to what we see with bradykinin, the physiological agonist. And this is NO, it is inhibited by nitroarginine, as expected. Now, we tested the effect of this blackcurrant extract on endothelial senescence in 2 different models. On the left-hand side is a replicative senescence model when you make passages of endothelial cells in our coronary artery endothelial cells very rapidly at P3. We already have 40 to 50% of endothelial cells in a senescent state. And you can see that this replicative senescence is inhibited in a concentration-dependent manner by the blackcurrant extract, here 3 to 10 microgram per ml, so need low concentration. And on the right-hand side, we also developed a premature model of senescence. We exposed endothelial cells to angiotensin II for 24 hours, and you can see S-beta-gal activity significantly increased. And here also, pre-treatment with blackcurrant extract was able to inhibit in a concentration-dependent manner the induction of senescence in response to angiotensin II. We also were able to confirm the induction of senescence looking at the small proteins, the senescence markers, which are inhibitors of the cell cycle. You can see that the angiotensin II induced upregulation of P53, P21, and P16 are all very nicely, strongly prevented by the blackcurrant extract. Now, what is the consequence if we prevent induction of senescence? This was tested here. We looked at the formation of nitric oxide using the fluorescent probe. In response here to bradykinin, we have an increased formation of nitric oxide. If now we pre-treat the cells, endothelial cells, with angiotensin II to induce premature senescence, you can see that the addition of bradykinin at the end had almost no effect. Those cells were no more able to produce nitric oxide. However, if we pre-treat the cells with the blackcurrant extract before applying angiotensin II, bradykinin is still able to stimulate nitric oxide formation. So those compounds are able to prevent the induction of endothelial senescence. Now, what is the mechanism? How do those anthocyanins interact with endothelial cells? This is a key and important question. And we stumbled in 2014 in this paper published by a group from China, where they actually look at delphinidine, which is delphinidine-3-O-glucoside. They looked at the protective effect of delphinidine-3-O-glucoside on oxidized LDL-induced apoptosis. You can see very strong induction of apoptosis, which was inhibited in a concentration-dependent manner by delphinidine-3-O-glucoside. And actually what they were able to show is that they were able to see uptake of anthocyanins using autofluorescence. In UVEX, you can see the red color is the autofluorescence of anthocyanins appearing in endothelial cells, and also in the isolated thoracic aorta, when it was exposed to delphinidine-3-O-glucoside, they were able to see uptake, the red color here, in the endothelium, in endothelial cells preferentially. And when they looked what is the mechanism, they were able to show that actually D-glucose was inhibiting the fluorescent signal, the accumulation of delphinine-thioglucoside. It was also inhibited by fluorescein. This is a non-selective inhibitor of sodium glucose cotransporters. And when they down-regulated expression of SGLT1 by using an siRNA, they were able to significantly decrease the accumulation of delphinine-thioglucoside, indicate that SGLT1 was important for the uptake of delphinine-thioglucoside. And we followed on this pathway using our blackcurrant extract. And we looked phosphorylation of enos in response to the blackcurrant extract, and we were able to show that actually a highly selective SGLT2 inhibitor, empagliflozin, had no effect. However, the dual inhibitor SGLT1, SGLT2, sotagliflozin, was able to fully inhibit phosphorylation of enos by the blackcurrant extract. And the less selective SGLT2 inhibitors, dapagliflozin and canagliflozin, also had an inhibitor effect. And you can see also on the right-hand side on the formation of nitric oxide induced by the blackcurrant extract, the highly selective SGLT2 inhibitor had no effect, whereas the dual SGLT1, SGLT2 inhibitor and the less selective SGLT2 inhibitor prevented NO formation, indicating that the sodium glucose cotransport, in particular SGLT1, is important for the uptake of conjugated anthocyanin into endothelial cells. And we further got evidence that indeed the uptake of blackcurrant extract in endothelial cells was decreased in a concentration-dependent manner by increasing concentration of D-glucose. Mannitol was inactive, so it's a D-glucose competitive event. And we can see on the right-hand side, decreasing D-glucose or D-glucose and sodium also decreased the accumulation of blackcurrant anthocyanins in endothelial cells. And then, of course, the question we asked, do endothelial cells express SGLT1 and SGLT2? We want to have direct evidence, because SGLT2 inhibitors are well known to protect the cardiovascular system. Lately, we have major clinical trials indicating protective effect in heart failure with preserved ejection fraction and also without preserved ejection fraction. And we looked at SGLT1 and 2 expression on the T cells using immunofluorescence staining. We were able to have a clear signal. And when we looked at the protein level, we were able to detect SGLT1 in the T cells. Here, a positive control kidney and intestine. And when we looked at the SGLT2 expression, we were able to see SGLT2 even in the T cells, which was completely unexpected, because it was, in the literature, it's in the kidney, the major organ where you see SGLT2 expression, but on the T cells also express SGLT2. And then we were interested, of course, to see, are all conjugated anthocyanins active in our model? And here we tested cyanidine-thioglucoside, beautiful uptake. We boost the autofluorescence using Naturstoff reagent A. And you can see the delphinine-thioglucoside also showed very nice uptake. The retinoside conjugated cyanidine and retinoside active, very poor uptake. So the uptake is dependent on the glucose, the type of sugar. Glucose is associated with a strong uptake. And this uptake, you can see, is inhibited by the dual SGLT1 and SGLT2 inhibitor, sotagliflozin. And here are the cumulative data. Now, senescent on the T cells is our target. So we looked what's going on in senescent on the T cells. And here you can see expression of SGLT1 in healthy endothelial cells, in senescent endothelial cells, and upregulation of SGLT1 expression in the pathologic endothelial cells. And here you see the data for SGLT2, low levels in the healthy endothelial cells, but a nice upregulation in the pathologic, in the senescent endothelial cells. And that's why we looked also at the uptake of blackcurrant extract here compared in P1 and P3, healthy senescent endothelial cells. And you can see the uptake in the healthy endothelial cells was even further increased. Occurs in a greater extent in the senescent endothelial cells. And you can see the sotagliflozin, the dual inhibitor SGLT1 and 2, had a significant inhibitory effect in P1 cells, but look what's happening in the senescent endothelial cells, very strong inhibition. And we looked also in what's the effect of the SGLT2 inhibitor, no effect of the uptake in P1, the healthy cells, but significant inhibition in SGLT2. And that's why we also looked what's happening in vivo. And we looked at SGLT1 and 2 expression actually in the aortic cross. You can see VCAM is upregulated because we have a site at risk, and you can see upregulation of SGLT1, upregulation of SGLT2, which then brings us to this conclusion that actually glucose-conjugated anthocyanins of cyanidin and delpidine appear extremely interesting to target endothelial cells which are characterized by premature senescence. And of course, we need further data to confirm that. I have a few more minutes? No. I will skip. We did an in vivo study in old animals regarding the effect of anthocyanins. I will show you very briefly. The old animals had hypertension, almost a high level of systolic blood pressure, which was significantly increased by the intake of blackcurrant juice. We were able to show that the blackcurrant juice intake is decreasing the hypercontractility of the arterial wall, reducing oxidative stress in the arterial wall in the old animal. And this is associated with a very nice improvement of the endothelial function. The overexpression of eNOS is reduced, the expression of P53, senescence marker, and activation of endothelial cells like by MCP1. Also, the local angiotensin system is decreased. In the old animal, you see ACE is overexpressed, AT1 receptor is overexpressed. Intake of blackcurrant juice has a very nice protective effect. So in conclusion, you can see that finally anthocyanins appear as interesting products. Again, we now have an idea that it's not all structure of anthocyanins. It depends on what kind of sugar is conjugated. And again, this overexpression of SGLT1 and 2 at arterial sites at risk makes it very attractive that we can target endothelial cells, the pathologic one, where we want to protect the cardiovascular system. And finally, to conclude, I would like to really acknowledge all the PhD students and the postdoc who did those beautiful studies, and our senior scientists in our international collaborations. And here's our new biomedical research center at Strasbourg University. Thank you very much for your attention.
Okay, time for one short question.
Yes, so
Sorry.
I'm up here. Yes, thank you very much for this presentation. My name is Roland Seifert. I'm the editor-in-chief of the Naunin Schmiedeberg's Archives of Pharmacology. And we got many, many hundreds of submissions on phytopharmacology each year. And I would like to take this opportunity to remind the pharmacological community, if you submit papers, make sure that in addition to an extract, you also include studies with purified compounds to elucidate the mechanism, as we have seen in the previous submissions. Otherwise, the papers will be immediately rejected. They simply don't fit into a pharmacological journal, and we see the great interest of the community, but we have to make sure that we look on mechanisms.
Okay.
Fully agree.
Okay, so, Valerie, thank you very much. And you need...
At least the title. Okay, now we will move on, and the next presentation will be given by Dr. Wen Zhang from Beijing, from the Institute of Materia Medicalis, and the title is Salvianolic acid A, a natural product from Salvia meteoriza bunge, alleviates cerebral ischemia, reperfusion injury via the inhibition of FoxO3a beam signaling pathway. Please.
Okay, thank you for the chair's kind introduction. It's a pleasure to be here, and it's my honor to speak before you at this pharmacology conference. Today, I would like to discuss the topic of stroke and the natural protective effect of salvianolic acid A for the treatment of ischemic stroke or cerebral ischemia reperfusion injury. My title of the topic today is Inhibition of FoxO3a beam signaling pathway, and MMP9 contributes to the protective effect of salvianolic acid A against the cerebral ischemia reperfusion injury. As we all know, stroke is a growing global burden, with being types of ischemic stroke, hemorrhagic stroke, and transient ischemic attack. Epidemiological studies have revealed that there are over 1 million stroke-related deaths worldwide each year, and ischemic stroke accounts for approximately 87% of all stroke cases. But so far, TPA is the FDA-approved thrombolytic agent for acute ischemic stroke, but with narrow therapeutic window and high risk of hemorrhagic transformation. Therefore, it is important to fund the new drug to treat ischemic stroke. The cellular and molecular mechanisms of stroke are complex. Multi-target mechanisms and cells participate in the pathological process of stroke, and the pathological stages and sequence of damaging events is complicated. Therefore, the development of multi-effects of drugs to treat ischemic stroke is become necessity. Then, in 2001, the scientists in United States proposed the concept of neurovascular unit. Nowadays, the protecting has shifted from solely protecting neurons to protecting neurovascular unit, containing the neurons, endothelial cells, microvessels, and glial cells, and some other supporting cells. As we all know, plants is a valuable source of medicines for centuries, and with nearly 200,000 natural products discovered to date. And China is one of the major discovered to natural products research. Natural medicines have been used in China to treat ischemic stroke for many years. And an analysis of 189 clinical trials involving 19,180 patients across 22 traditional formulations evidenced the effectiveness of the traditional Chinese injections in improving patients' neurological deficit. However, some drugs need further validation of their therapeutic effect and validation of their action mechanism in order to be approved abroad. Danshen is a medical herb widely used in traditional Chinese medicine. Modern scientific research have revealed that danshen has a variety of pharmacological activities, including antioxidation, antiplatelet aggregation, protecting the cells from apoptosis, inhibiting ischemia reperfusion injury, promoting the endothelial cells, acting on lung channel functions, anti-inflammation, and so on. Salvianolic acid is an active natural product extracted from danshen. Previous mechanism studies of salvianolic acid in stroke models have indicated that salvianolic acid could downregulate RS, inhibit soluble epoxide hydrolyze. Other disease models also indicated that salvianolic acid has a variety could regulate a variety signaling pathway, including activate of AMPK PGC1 alpha SIRT3 pathway, and downregulate the phosphorylation of JNK signaling pathway, upregulate the phosphorylation of ERK, and activate the BMPR2 and the SMART signaling pathway, activate NERV2, upregulate the NOS, inhibit the AGE and the RAGE signaling pathway, and so on. So in this study, we will first confirm that salvianolic acid could the protective effect of salvianolic acid on blood-brain barrier and neurovascular unit by inhibit MMP9, and then we will confirm that salvianolic acid I could inhibit cell apoptosis by promoting the phosphorylation of FoxO3a. So first, in vivo study, we used the right MCL reperfusion injury to elevate the salvianolic acid, the therapeutic effect. The result showed that MCAO reperfusion injury could increase the right neurological deficits and significantly increase the right brain water content. Well, salvianolic acid treatment could reduce the neurological deficits and reduce the brain water content in a concentration-dependent manner. TTC staining was used to assess the right brain effector volumes. So MCAO reperfusion injury will significantly increase the effector volumes. Well, salvianolic acid treatment could significantly reduce the effector volumes in a concentration-dependent manner. Then, HE and nasal staining show that salvianolic acid could elevate the brain injury in IR rats. In addition, salvianolic acid could inhibit the oxidative stress, downregulated the MDA production, upregulated the SOD and GPX levels. For further study, we focused the protective effect of blood-brain barrier. As shown in Figure A, MCL reperfusion injury would increase the immense blue elucidation and severe result in tight junction proteins degradation, and the salvianolic acid treatment could inhibit the immense blue elucidation and inhibit the tight junction proteins aggregation, including the protein levels of CL1, claudin 5, and aclaudin. For further Western blotting, studies have revealed that MCL reperfusion injury would increase the protein levels of MMP9 and MMP2 and decrease the protein levels of TIMP1. Well, salvianolic acid treatment could inhibit the IR injury-induced MMP9 upregulation and inhibited the TMMP1 downregulation. Then the MMP2 and MMP9 expression were detected by the glytin-thermographic assay, as shown in Figure D. Salvianolic acid treatment could significantly downregulate the expression levels of MMP9, but has no significant effect on the MMP2 protein levels. So our in vivo study verified the therapeutic effect of salvianolic acid. The further study we will focus on the action mechanism of salvianolic acid in vivo and in vitro study. First, in vitro study, we use the OGDR stimulation. The OGDR stimulation will result severe cell damage and cell viability loss. Well, salvianolic acid treatment could inhibit OGDR-induced cell viability loss, as shown in MTT assay and under the inverted microscope in a concentration-dependent manner. Well, in vitro study, the 5 micromole concentrations of salvianolic acid showed the maximal protective effect of cell viability loss, so it was selected for further study. As we all know, FoxO3a regulates the cell apoptosis, and OGDR stimulation downregulated the phosphorylation of FoxO3a and AKT, while salvianolic acid-A treatment could upregulate the phosphorylation of AKT and FoxO3a. As shown in Figure C and Figure D, OGDR stimulation also downregulated the phosphorylation of mTOR and GSK3-beta, but salvianolic acid-A treatment could not have no significant effect on the protein levels of mTOR and GSK3-beta. So further study was focused on the regulation of FoxO3a by salvianolic acid-A. In addition, OGDR injury also upregulated the protein levels of BIM and cleaved caspase-3, which represents the cell apoptosis. While salvianolic acid-A treatment could downregulate the protein levels of BIM and cleaved caspase-3 in a concentration-dependent manner. The above results indicate that PI3K, AKT, and the FoxO3a signaling pathway contributed to the neuroprotective effect of salvianolic acid-A on cell apoptosis. So to find out whether the phosphorylation effect of FoxO3a and the AKT the 5 micromole concentrations of salvianolic acid-A were added and incubated for 1, 2, 4, and 6 hours. And different concentrations of salvianolic acid-A were pretreated for 4 hours. The results indicate that salvianolic acid-A could increase the AKT and FoxO3a phosphorylation in a time and concentration-dependent manner. So the above results showed salvianolic acid-A could increase the phosphorylation of FoxO3a and AKT. Then to find out whether the PI3K AKT signaling pathway could contributed to salvianolic acid-A induced the FoxO3a phosphorylation. The PI3K inhibitor was used. And firstly, the cells were pretreated with the PI3K inhibitor for 40 minutes before the cells were subjected to OGDR stimulation and the salvianolic acid-A treatment. Then the MTT assay was carried out. As shown in Figure A, the pre-incubation of the PI3K inhibitor could reverse the salvianolic acid-A induced cell neurological effects. And as shown in Figure B and Figure C, the pre-incubation of the inhibitor would reverse the salvianolic acid-A induced the phosphorylation of AKT as well as FoxO3a phosphorylation. As shown in Figure E, salvianolic acid-A treatment could inhibit the nuclear translocation of FoxO3a, which was also reversed by the interference of PI3K inhibitor. Then, to evidence the FoxO3a beam signaling by interference the FoxO3a, the FoxO3a siRNA were introduced. As shown in Figure E, OGDR injury could upregulate the nuclear FoxO3a levels, while salvianolic acid-A treatment could downregulate the nuclear FoxO3a levels, which was partly reversed by FoxO3a siRNA, which triggered the downregulation of protein levels. So these results show that PI3K AKT mainly contributed to the salvianolic acid-A induced the phosphorylation of FoxO3a in SY5Y cells, and the phosphorylation of FoxO3a would downregulate the nuclear FoxO3a levels, which further downregulated the protein levels of BIM. The above in vitro study, we studied the action mechanism of salvianolic acid A, that salvianolic acid A could inhibit cell apoptosis via PI3K, AKT, and the FoxO3a signaling pathway. Well, if salvianolic acid A had the same effects and mechanisms in vivo study, we also used the right MCO reperfusion models. As shown in Figure A and Figure D, the MCO reperfusion injury could downregulate the phosphorylation of FoxO3a and AKT in the right brain cortex region, and the salvianolic acid A treatment could inhibit the downregulate of FoxO3a and AKT phosphorylation. In addition, the upregulate phosphorylation of FoxO3a and AKT was reversed by the interference of PI3K inhibitor. And as shown in Figure D, MCAO injury also downregulated the phosphorylation levels of FoxO3a and AKT in right brain hippocampus region, and salvianolic acid A also could increase the phosphorylation levels of AKT and FoxO3a, which was also reversed by the inhibitor treatment. Then, in immunofluorescence assays showed that salvianolic acid A could inhibit the nuclear translocation of FoxO3a in neurons, both in right cortex and hippocampus region, which was also partly reversed by the inhibitor of PI3K treatment. In addition, MCO reperfusion injury also upregulated the expression of BIM and collude caspase 3, which represents the cell apoptosis in right cortex and hippocampus regions. And the salvianolic treatment could significantly inhibit the expression levels of BIM and collude caspase 3 in a concentration-dependent manner. So the TO staining was assessed the cell apoptosis rate, as shown in Figure E and as shown in Figure A and Figure C. MCO reperfusion injury model could significantly increase the cell apoptosis in neurons, both in cortex and regions, and the salvianolic acid A treatment could significantly inhibit the neurons cell apoptosis rate, which could reversed by the PI3K inhibitor. And as shown in Figure E, MCO reperfusion injury could downregulate the cell, could inhibit the cell apoptosis of GLUT1 positive cells. Well, salvianolic acid A treatment could inhibit the percentage of GLUT1 positive cells apoptosis, which was also reversed by the PI3K inhibitor treatment. And the TTC staining was used to assess the right brain infect volumes. As shown in Figure J, there was almost no infect volumes in sham group, and MCO reperfusion injury result in severe infect volumes compared with the sham group. Well, salvianolic acid A treatment could significantly downregulate the percentage of right brain infections, and the treatment of PI3K inhibitor would partly reverse the salvianolic acid A effect of the brain infections. Well, MCA or reperfusion injury also result in the neurological impairment. With the same group, while salvianolic acid treatment could downregulate the right neurological deficits, which was also could partly reversed by the PI3K inhibitor. So in our in vivo and in vitro studies, we also verified the therapeutic effect of salvianolic acid A and the action mechanism of salvianolic acid A in MCO reperfusion injury and OGDR reperfusion injury. In conclusion, salvianolic acid A could elevate the cerebral ischemia reperfusion injury at least partly via the AKT FoxO3a and the beam signaling pathway, and protecting effective blood-brain barrier and neurovascular effect by inhibit the MMP9. This is all my report. Thank you for your attention.
Thank you very much for this beautiful study. This paper is now open for discussion. Yes, please. I don't think the microphone is working. Closer.
Do you have any data?
No, not working.
Sorry, I can't hear.
Just need to speak close to the mic, please.
Do you have any data to support that this compound work as neurogenesis also? Any data to support neurogenesis in a stroke model?
Is your compound working on neurogenesis, is the question.
The salvianolic acid is an active natural product from dantian, and another natural product, and dantian has also another active natural product like salvianolic acid A, B, C, D, E, F, and so on. But salvianolic acid A is discovered by my supervisors and have studied for many years. And we have the confidence that salvianolic acid A is a successful new drug for treat the ischemic stroke. And we use the middle cerebral artery occlusion and the oxygen growth OGDR stimulation, these models to find out whether salvianolic acid could have the therapeutic effect.
Thank you. Any further questions? Very short question, short answer.
Yeah, just a quick question. I will read from here. So what is known about the bioavailability of salvianolic acid? Because I guess dantian is used orally in China. So any clue about bioavailability of this type of compound? If it gets metabolized, or because in your assay you show very nice in vivo data, I guess the compound was even IV.
Yeah, the traditional dantian is a medical herb widely used to treat ischemic stroke for in China for many years, and and has always made the traditional Chinese injections. And
Done by injection.
Yeah.
Thank you very much. We now have to move on because time flies. Thank you, and I would like to introduce the next speaker, which is Dr. Xiaohua Lu. She comes from the University of Mainz in Germany, and she's part of the team of Thomas Effert, and her title is Network Pharmacology of Angiogenesis Inhibitors Inspired by Nature. Please.
Okay. Hello, everyone. First of all, I would like to express my appreciation for the kind invitation and also introduction. So it's really a great honor and pleasure for me to share our works. Today, the topic will be network pharmacology of angiogenesis inhibitors inspired by nature. So what is the network pharmacology? In the recent years, the establishment of the series of biologic database have been developed for the to support the pharmacology research for a certain disease. The database such as GeneCart or OMI could be used to identify what kind of genes could be used to could be identified to figure out what kind of genes could contribute to the disease. For a specific compound, the database such as the Swiss target prediction and PharmaMapper could be used to figure out what kind of genes could be affected by this compound. In this way, a set of common genes could be easily obtained to construct the disease's phenotype gene target drug interaction network. The following database could be used, such as the strain cytoscape and the ingenuity pathway analysis, as well as the in silico techniques. Then further studies, both in vitro and in vivo, could be used to validate this effect of this specific compound on the diseases network. Cancer is the second leading cause of death worldwide after cardiovascular diseases. It's estimated 19.3 million new cancer cases and around 10 million cancer cases in 2020. A huge amount of the money has been used to support for the cancer treatment. With the development of the medical research, a series of anti-cancer strategies have been developed. Even though cancer is still the most stubborn disease in the world. So you may ask, what are the causes of cancer? Why they are so difficult to overcome? Generally speaking, cancer is a genetic disease. It is caused by the genes that control the way our cells grow, divide, and function. There are three types of genes: oncogenes, tumor suppressor genes, and the DNA repair genes. The changes of these genes could help the cancer cell to grow out of control, to invade the growth suppressors, and to activate the invasion and metastasis, as well as to induce the angiogenesis. Inhibitors of this changed genes or the proteins could be the promising anti-cancer agents. As we can see here, inducing angiogenesis is one of the main hallmarks of cancer. Inhibitors of VEGF signaling pathways could be the promising anti-angiogenesis strategy. So what is the angiogenesis? As the tumor grows, it eventually reaches a size where it requires additional vasculature to sustain continued growth. To reach this goal, the tumor cells secrete certain proteins to promote the blood vessel growth around and into the tumors. This process is called angiogenesis. Meanwhile, the cancer cell can transfer to other sites of the human body through the blood vessel to form the metastasis tumors. Now let's take a closer insight into the VEGF and their receptors. VEGF are the main drivers of angiogenesis. This family includes 5 members: VEGF A, B, C, D, and placental growth factors. VEGF bind to their receptors, leading to its phosphorylation and also the formation of the hemodimers and heterodimers. VEGFR includes 3 parts: extracellular domain, transmembrane domain, and the kinase domain. VEGFR1 mainly responsible for the development of hematopoietic cells and the migration of the monocytes and the macrophages. VEGFR2 mainly responsible for the angiogenesis, migration, proliferation, and survival of endothelial cells. VEGFR3 mainly responsible for the development of the lymphoendothelial cells. So VEGFR2 play a dominant role in the angiogenesis. In the market, we can easily find the anti-VEGF or the anti-VEGFR2 inhibitors. However, they display the side effects such as the hypotension, thyroid dysfunction, and cardiotoxicity. Therefore, it's necessary to look for the novel anti-angiogenesis inhibitors. Lateral products have historically played a key role in the drug discovery, especially for the cancer and infection diseases. In the latest 40 years, almost 50% of the FDA-approved drugs are derived from the lateral products. Recently, we wrote a review which summarized the anti-angiogenesis natural products. In this review, we summarized around 150 natural products. The underlying mechanism for these natural products is that they could inhibit the VEGFR2 signaling pathways, thus to inhibit the vascular permeability, cell migration, and the cell proliferation. In our lab, we are specifically interested in the artemisinin type compounds. As we all know, for this type compounds, they have been developed as the standard therapy for the malaria treatment, while the biologic activities for the artemisinin type compounds are not limited to the malaria treatment. These type compounds, they could also have the effects on various diseases. Such as the viral infection, schistosomiasis, trypanosomiasis, and so forth. The most important part is that the artemisinin type compounds could inhibit the cancer. Not only in our lab, but also in other labs, we proved that the artemisinin type compounds inhibit the cancer through multiple ways. It could arrest the cell cycle, and it could also to induce the apoptosis and to inhibit the angiogenesis. So our question is there any artemisinin type compounds which could target VEGFR2, therefore to inhibit the angiogenesis? This is our project goal. As an initial step, we performed the clinical relevance to figure out the prognostic potential of VEGFR2. In the second step, we performed the molecular docking to look for the most potential artemisinin type compounds which has the favorable binding affinity with VEGFR2. In the last step, we performed both in vitro and in vivo studies to validate the anti-cancer mechanism of the selected artemisinin type compounds. And now let's come to the results part. Here is the prognostic potential of VEGFR2 in different cancers. We applied the PrognosScan to check the survival rate of the cancer patients with high or lower expression of VEGFR2. The Kaplan-Meier plot indicates that the higher expression of VEGFR2 in breast cancer patients is correlated to a lower survival rate. This is also observed in colorectal cancer, esophagus cancer, eye cancer, lung cancer, as well as the overall cancer. In the following slides, I would like to introduce two artemisinin type compounds which were reported in our group last year. These two compounds has showed a favorable anti-angiogenesis effect. The first one is the FOES123. How this compound was selected out? We obtained 52 and 364 artemisinin type compounds from the PubChem and PubMed respectively. In the multi-docking, here is the bar chart for the number of the artemisinin type compounds in response to different category of the binding energy with VEGFR1 or 2. The reference compound artesunate showed a binding energy at around minus 9.7 kilocalorie per mole. 194 and 245 artemisinin type compounds showed a lower binding energy with VEGFR1 or 2 in comparison with the artesunate. It means that most of the artemisinin type compounds had a good binding affinity with VEGFR1 or 2. This is the linear correlation between the lowest binding energy and the predicted inhibition constant. It means that this molecular docking method is well established. In the most interesting point in this part is that the top 10 artemisinin type compounds bind to the similar space of VEGFR2. 4 of them are commercially available, so they are used for the further experiments. FOES123 was finally selected out because of its strong cytotoxicity on different cancer cells. Here, the breast cancer cell line, MDAMB231, is the most sensitive solid tumor cells with the IC50 value around 10 micromolar. The leukemia cells, CCRFCM and CEM-ADR5000, showed even higher sensitivity toward this compound. The IC50 value for the peripheral blood mononuclear cells was around 84 micromolar, which means that this compound had a meaningful effect on the human healthy cells. When we look back to our molecular docking result, we found that FOES123 could be stably located at the ATP binding pocket of VEGFR2. A list of the 18 residues are responsible for this effect. Human benign vein endothelial cells, Huyck cells in short, is a commonly used cell model to study the angiogenesis function of the endothelial cells. Here, this compound could inhibit the cell viability of Huyck cells at around 4 micromolar. In the presence of VEGF, the cell viability of Huyck cells is increased around 15%. FOES123 had less effect on the cell viability of the VEGF-induced Huyck cells. Here is the tube formation result. VEGF could enrich the formation of the capillary-like tubes. while FOERS123 could disrupt the tube formation as evidenced by the sharp decrease of the branching point and the turbulence. Cell migration is an initial step for the cell invasion. Both the cell migration and the cell invasion are dependent on the tumor angiogenesis. Here, we performed we performed the 2D wood AC and the 3D spheroid AC on the most sensitive solid tumor cells, MDA-MB231 toward FOERS123. As we can see here, FOERS123 could inhibit the cell migration on these both models in a time and dose-dependent manner. Likewise, FOERS123 could suppress the cell invasion in a dose-dependent manner. Here is the apoptosis. Usually, cell apoptosis is carefully regulated in the human healthy cells, while for the cancer cells, due to the genetic changes, the cancer cell can not only grow out of control, but also to lose function of the apoptosis. Here, we performed the cell apoptosis using the flow cytometry. This diagram shows the different cell population. Once the cancer cells undergo the apoptosis, the cell population would shift from the right to the left and the top. As we can see here in the bar chart, the cell apoptosis was significantly induced by this compound FOERS123. To have a real interaction between FOERS123 and VEGFR2, we performed the microscale thermophoresis experiments. As we can see here, this curve, it means that FOERS123 could strongly bind to the VEGFR2 with the dissociation value at around 1.3 micromolar. To further check the relevant signaling pathways, we also performed the Western blot. We observed that not only the phosphorylated VEGFR2, but also the phosphorylated GNK, ERK, and AKT were reduced. Chloreal anatoic membrane assay, CHEM-assay in short, is a commonly model to screen out the angiogenesis agent in the pharmaceutical research. This method is quite easy, inexpensive, but highly reproducible. So in this experiment, FOERS123 inhibited the formation of the blood vessel, and also to inhibit the formation of the branching points. That is all the results regarding our first compound, FOERS123. The second compound is FO8643. Yes. For this compound, since we applied the seminal techniques and methods, I will not go into so much detail. Here, I just give you an overview or a summary. For FO8643, we also tested the cytotoxicity on different cancer cell lines. The liver cancer cell line HUH7 was the most sensitive solid tumor cells. This compound could also arrest the cell cycle and the G2M phase. This FOS643 could also induce substantial apoptosis in microarray AC and the angiogenesis AC. This compound could also inhibit the cell migration and also the angiogenesis. That's all the preliminary experiments we have done so far. But their story about these two compounds will not stop here. For the next step, we are also planning to study the following perspectives. So we would also like to investigate the correlation between cancer stem cells and angiogenesis to check the metastasis mechanism and the anti-tumor activity as well as the side effects. Okay, so I am really thankful to have the CSC grant to join Proof Effort group, and thank you for your attention. Now I'm open to your questions.
Thank you very much for this very nice presentation, which is now open for questions. Yes, please.
Yes, this is a wonderful work you did in the Effort Lab. And Thomas Effort is one of the editors dedicated to the natural compounds. And this is a wonderful study where you do network pharmacology not only in silico but with real pharmacological experiments, because there is now an inflation of submissions just with a virtual docking, and this is not really useful for. You really need experimental validation, so congratulations to this wonderful work.
Yeah, thank you for your for a question.
Yes, yours.
Geneva, just the second the compound that you mentioned briefly, what is the IC50? Because the first one was 1.3 micromolar, which is okay, but not great. And how much is the second one?
For the second one, actually, we also, because of the time limitation, I didn't put so much detail. For the second compound, F08643, we also performed the resuscitation-based cytotoxicity assay on different cancer cell lines, like the leukemia cells and the solid tumor cells. For the solid tumor cells, it's around 10 to 20 micromolar. Yes, and for the suspension cell line, such as the CCRF or CCRF CEM or CM80R5000, it's even much more lower. It's around, if I remember correctly, it's around 0.3 micromolar. If you need more details, you can also refer to our publications. Yeah.
Okay.
Yeah. Thank you.
Thank you.
Yes, please.
I think I appreciate your presentation and the data. I have a couple of queries. Actually, we have done studies in 2006, artemisinin compound in experimental induced colon cancer, and it was found to be very, very efficacious. So when we carry on that work, we had only challenges because this is a chronic therapy. You have to give it for a long period of time. And challenges it comes that it also affect the brain, artemisinin compound. So how do you address the issue tomorrow when you go to clinics that it is going to affect the brain in terms of seizure-inducing agent? So how do you address in terms of the dose selection and effect on the CNS? Thank you.
Okay. Thank you for your question. Actually, this is really a good question regarding the artemisinin type compounds dose selection. Even though we didn't, we don't perform any dose information on the in vivo studies so far. As you can see in our presentation, we only performed the chemAssay. But as you know, for the in vivo studies, like the rats or the monkeys, this would be the preliminary in vivo studies. And like when we talk about the clinical studies, I think we need to put more effort on this. Yeah. I hope I answer your question. Yeah.
One last question, please.
Very nice presentation. My question is almost same. What's the status of artemisinin in other types of carcinomas apart from breast cancer in which we have studied?
Sorry, I didn't really get your point. Could you please?
can we use this artemisinin derivatives in other types of cancer? We have studies on breast carcinoma, na?
If I understand you correctly, you mean if this this artemisinin type compounds can be applied to the breast cancer?
Other types of other cancers, other carcinomas, can we?
Based on our first experiments, the resistance-based cytotoxicity for this type compounds, they do have different. They do have the cytotoxicity on different cancer cell line. So for the breast cancer cell line, MB231 was the most sensitive one. I believe, like, since we just use a limited number of the cancer cell line, even though this cell line from different cell type, but I think we need to test more cancer cell line to check the possibility or the potential of this artemisinin type compounds and the cancer.
Right. Thank you.
Thank you too.
Thank you very much. No more questions? Then thank you, Dr. Lu, and thank you. And we now will conclude the session. I would first of all thank all the speakers for a wonderful presentation. I think we are convinced that from the phytochemistry level up to preclinical and clinical data, there is potential for natural products, but we need very good characterization, identify the active pharmacophore, and of course also look at the end for toxicity. And thank you very much for the audience for contributing, and I give the last word to Professor Spedding.
I think it's been actually a great day for me as well, because I started to look for, as Secretary General of UFAR in 2014, to advance the field, and I think you can see just how far it's gone. And it is getting quite a long way. There's an issue I'd like to bring up for the editors, that Dr. Wolfender has clearly shown a way forward to ensure we can see which compounds are going to be active and which compounds are going to be toxic, and whether these compounds are acting in synergy or not. So one of the main reasons I wanted to start this was to prevent people just putting mixtures into animals and it not going any further. And I think now we're at a stage whereby we can begin to bring it together so we can see groups of compounds now. I think that's scientifically valid to be published. So I think that's a point I would like to make. And I think I would just like to thank everybody again, and my co-chair and worked together for ages, Valerie. And so thank you for a great seminar.
Thank you. We close the session. Thank you.